Welcome everyone to the EEGNet Spring Colloquium. We'll let everybody get settled and Alan Evans will begin the welcome remarks in just a minute.
Good morning, everybody. My name is Alan Evans. I'm the Scientific Director of the EEGNet network, so welcome to our Spring Colloquium. I'm joined in coordination of this event by my colleague within the Global Brain Consortium, Pedro Valdes-Sosa. You'll hear today about both the national EEGNet network and the international Global Brain Consortium. We have a busy schedule today. I will make some opening remarks, and then we'll have some words from our philanthropic donor and beneficiary of the EEGNet network, Irving Ludmer. We're also funded by Brain Canada. We'll hear from Sabina Antonescu, who represents Brain Canada. And then, as you see on this slide, we have a series of talks from Pedro and Christine and Jorge-Bosch Bayard speaking about methods development in particular. Then Sid Segalowitz, Philippe Albouy, and Sarah Lippé, and Faisal Mushtaq will be talking about applications built on top of these networks. I'll make a few introductory remarks because there are plenty of people in this symposium who haven't been here in the past, so I apologize to those who've heard some of this before. So this network is the EEGNet symposium, and we're looking at global open EEG standards built on top of the EEGNet platform and the supporting ecosystem. We invite your participation to contribute data and tools, and we also have annotation working groups associated with EEGNet. If you have questions, please place those questions in the chat box for the Q&A session at the end of this 2-hour period. We have many upcoming initiatives associated with EEGNet and the Global Brain Consortium. For instance, in June at the OHBM meeting in Seoul, we have an educational course entitled Global Open Science Electrophysiology. In September, we have the Global Brain Consortium annual meeting, and we have numerous initiatives which are extending out from EEGNet and the GBC. Remember that the EEGNet network is built on top of the Canadian Open Neuroscience Platform, CONP, so that's mentioned here. We also have numerous initiatives in play within the US arena. The Healthy Brains Child Development HBCD network, the activities going on here are very much part of that large US study to explore opiate addiction in children. We have numerous collaborations in the development of electrophysiological annotation and scoring systems with the NAMR team of Scott McCaig. And of course, we have a methodological work to improve the EEG bids format. EEGNet is made up of 35 Canadian PEIs from 6 provinces across the country. The funding partners include Brain Canada, as already mentioned, and Professor Irving Ludmer. Also have University of Laval with Philippe Albouy representing the CERVO network, and Brock University, Sid Segalowitz. I've already mentioned that EEGNet is built on top of the Canadian Open Neuroscience Platform. I'll be showing you a few slides showing you what that linkage is. And then we have the partnership with the Global Brain Consortium. Just a reminder of the goals of EEGNet as funded by the Brain Canada grant. It was to create a unified platform for sharing and analysis of standardized EEG data sets to the national and international research community. The outcomes are a gold standard expert annotated open EEG data sets hosted via the EEGNet platform, ready for machine learning, clinical applications, and early identification of biomarkers. On the right, you can see the original goals of the EEGNet grant and the progress that has been made. As you see, I will not go into this in detail because Christine Rogers is going to go into this slide in more detail, but I think the general point to be made here is that most of the deliverables for EEGNet have been delivered, and there are some outstanding ones shown by the orange cones, which are still to be done. We're in the midst of closing those initiatives down, but I think in general, you can see that we've made good progress on our goals. The Canadian Open Neuroscience Platform is also funded by Brain Canada, and this brought together many sites across the Canadian scene. It involved technical integration, international partnerships, ethics and data governance of data sharing across both national and international boundaries, and analysis packages. As you can see here, the CONP portal at this point has approaching 100 data sets and approaching 100 workflows and pipelines which have been set up to run on the CONP infrastructure. What is the CONP infrastructure? In particular, it is made up of two platforms. The LORIS multimodal databasing platform on the left of this slide, which allows us to integrate imaging, genetics, epigenetics, behavioral clinical data, biospecimen, tissue samples, and electrophysiology all under one roof. And that's very important for many large projects such as HBCD that I mentioned, because they have many different forms of data to be collected. So LORIS underpins those multi-site, multi-time point, multimodality projects. CBrain, in essence, is a portal to the high-performance computing infrastructure, and originally of the country and now internationally, as well as cloud services. As you can see here, the CBrain incorporates at least 3 of the XSEED sites in the US, as well as supercomputing infrastructures in Europe to go alongside the Canadian sites. There are now about 1,500 users internationally making use of the CBrain platform in 32 different countries. One new development since the last time we talked is that this platform infrastructure has been commercialized by the company Lasso Informatics, which, as you can see here, allows us to incorporate all of those different data types under one roof. And it's notable that this platform has been recently adopted by the National Institutes of Health to provide a no-compete contract to support the dissemination of large cohort data sets like HBCD and its progenitor, the ABCD, Adolescent Brain Child Development Network. I think this is notable that this is the NIH in the US has adopted essentially a startup Canadian company, which is making use of the same infrastructure that EEGNet and GBC use. Last slide, these are some of the metrics in terms of the data that EEGNet is making publicly available: data, tools, the user community, and publications. That's all I need to say at this point, because there's much more to come in more detail from our speaker list. I do want to now make time for some words from Irving Ludmer, of our benefactor. Thank you very much.
Bonjour, hi, hola. My name is Irving Ludmer, and I lend my name to the Ludmer Center for Neuroinformatics and Mental Health. This center has been built on the idea of collaboration, transdisciplinary approaches to neuroscience and open science, with the ultimate goal to accelerate discoveries and treatments for mental health and brain diseases. Since its founding in 2013, the Ludmer Center for Neuroinformatics and Mental Health has been a proud supporter of initiatives like EEGNet and the Global Brain Consortium and the Canadian Open Neuroscience Platform. EEGNet has brought together partners from across Canada and across the world to realize the vision of global standards in EEG with online interactive tools, making virtual collaboration possible as never before. I'm delighted to witness these collaborative efforts, advancing our understanding of the brain and unlocking the potential of neuroscience technology for applications around the world. I wish to congratulate the EEGNet team and network of research collaborators who have built an impressive open science neuroinformatics platform demonstrating the potential for transformative impact in global health and clinical research. And I wish to thank EEGNet's partner institutions and Brain Canada for ensuring the continuity of these collective efforts and their continuous support of our dedication to accelerate discovery in brain research. Thank you for coming, and we hope you enjoy the EEGNet Colloquium.
Christine?
Thank you. Now we have remarks from Sabina Antonescu from Brain Canada. Welcome, Sabina.
Thanks, Christine. Hi, everyone. My name is Sabina Antonescu. I'm the Program Operational Lead at Brain Canada. I'm thrilled to join you all today and really look forward to the presentations and discussion to come. It has been wonderful to see all the progress that has been made in the last few years, and I really want to commend the EEGNet team, the collaborators, partners, everyone for all that you've achieved thus far. Not only in the development and launch of the platform, but as well the innovative ways that researchers have been utilizing the platform and the tools and data that are available in the context of their research. As mentioned by Dr. Evans, Brain Canada's support of EEGNet and the CONP has been made possible through our signature Platform Support Grant Program. I'm pleased to announce that our next Platform Support Grant Program will be launched shortly with 2 streams available for funding, one for new platforms and one available to previously funded platforms by Brain Canada. So I encourage you all to keep an eye out for this announcement in the coming weeks on our website and our communication channels. And I hope to see some applications come through from the team. So thank you again for having me, and yeah, looking forward to it.
Thank you very much. Now we move to opening remarks with Pedro Valdes-Sosa.
Hello, can you see my screen?
We can see your screen great. Yeah.
Excellent. Thank you.
It's very quiet, Pedro.
It's very quiet. Let me talk a bit louder then. First, thank you all for coming to this symposium of the EEGNet. This is a very important moment since now I think we're ready for prime time. And my task now is to explain what is the background, why we want something like this. And this is best explained by talking about what the Global Brain Consortium is and what it's the ideas that it embodies are based upon. So in the first place, I would like to mention that I'll be showing some documents you can look at in greater detail. This is the latest analysis of the global burden of brain disease that at this moment is now talking about disorders affecting the nervous system. And it's a systematic study in which one of the Global Brain Consortium members and EEGNet members, which is Maria Luisa Bringas, participated. Here, we have a breakdown of the global burden of disease. We're going to share the screen, we're going to share the presentation, and practically what one can see here is the huge amount of disability life-adjusted years that are caused by brain dysfunctions. But the important problem is that they're disproportionately impacting low-income and middle-income countries. These are the places where access to treatment and rehabilitations remain very limited. And this is the inspiration for the work of the Global Brain Consortium, which in fact was motivated by this meeting in 2016 at the headquarters of the World Health Organization, in which the leaders of the global brain projects met And the conclusion was reached that the amount of funding available for brain research worldwide did not actually match with the impact on the global burden of brain disorders. This has been elaborated, and this is the editorial of a special issue of Neuroimage on neuroimaging and global health, since one of the limiting factors is the availability of technology. This leads to the realization that if one wants to change this situation, one has to use the appropriate technology. And of the many imaging technologies which all are necessary, and which the Global Brain Consortium is also engaged in, it is electrophysiology, what we see here, which is neuronal activity, which has the fastest time response and is the most cost-effective. For that reason, we have concentrated our efforts on electrophysiology, and EEGNet, together with GBC, has analyzed that the use of electrophysiology has several dimensions. And one is, what is the status of health of people that go from normal to abnormal? The other is which stage or which level of the health system it is engaged in. And then the other is, what is the state of the lifespan? So it's really a multidimensional problem. In there fit different disorders and even brain health, which is a concern now. Now, basically, one of the things that we are interested in is how neuroscience can affect the global burden of brain disorders. It is through neurotechnology and diagnosis and treatment, but only if we reach from the tertiary level to the primary health level. Now, to be able to do this and reach out, we need to have instruments. And the way we see it is that electrophysiology plays a particular role in what could be active screening to provide population brain health across the world. For that, we need instruments. But this is not a dream. Cuba, particularly the Cuban Center for Neuroscience, has shown that even in countries with health systems that are not developed, one can use electrophysiology, for example, for hearing screening, and screen hundreds of thousands of children in a few months. Now, this requires the appropriate technology in EEG and electrophysiology, and we're thinking of EEG, including evoked potential, spontaneous activity, and that is precisely the concern of what we're promoting. And also, these standards have to be shared. So for this reason, the Global Brain Consortium was created in 2019. Here we see the inaugural session in Montreal. We see the countries that joined, and the purpose of the Global Brain Consortium is to promote the development of tools and techniques and for the analysis of EEG and the sharing of data. This is the steering committee, which has members of many of the different brain projects of the International Federation of Clinical Neurophysiology. And our strategy is to develop quantitative methods for EEG, where once again, I say we're including evoked potentials. It is to have federated databases that can then share either the data, the raw data, or processed data, and then to use neural modeling and all of this to inform artificial intelligence method to be able to carry out what we call precision quantitative EEG, which is individualized and personalized diagnosis of different brain dysfunctions. The basis of all this is the availability of the Canadian Open Neuroscience Platform, and Alan has always referred to the storing of data, of longitudinal data and LORS, but also the access to all the algorithms through CBrain. Data that is in the federated databases, for example, the data of the Cuban Human Brain Mapping Project, where we have EEG, MRI, there is a psychological test, clinical data, the blood is stored, and this was published in Nature Scientific Data a number of years ago. I want to really emphasize that this data will increase, and the annotation of this data is being done in collaboration with the International Federation of Clinical Neurophysiology. One of the upcoming meetings in September is the Congress in Jakarta of this organization, with whom we have an agreement for promoting the use of advanced EEG technologies. In particular, we have focused, because it's very standardizable, with quantitative EEG. Here's the references. We now have, from all these countries, which is 9 countries, a federated database where we have achieved normative data from age 5 to 100, which is harmonized, and where we have eliminated the effect of equipment and local effects, let's say, that may be due to some country-specific characteristics. There's a paper out in the name of the Global Brain Consortium on this, and of course, these norms have been shared, and the characteristic they have is that they're norms not only for, let's say, EEG spectral activity, but also for functional connectivity. We now are working with the EEG Many Labs initiative, and Faisal is one of the main proposers of this, together with Yuri, and this will increase, together with other groups, the database. This will expand it. Here we see the new databases, but we're working specifically with Africa, which is, let's say, the big blue zone there, and working with 4 countries that have it incorporated into the Global Brain Consortium. This is the software that later will be explained, which is openly available for spontaneous activity. We have been able to carry the voice of the Global Brain Consortium to WHO. So we've participated, Alan and I have contributed to the declaration on what brain health is and what are its objectives worldwide. And this is a particular meeting to discuss how to measure at population levels the development of children. And very relevant to this, and it's information that's in that report that just came out, is the work we've done in malnutrition in Barbados, in which we've been able to follow up for 45 years and see the effect of a single episode of malnutrition in the first year of life over the lifespan. And here I'll mention a particularly recent result published in Human Brain Mapping just a few weeks ago, in which we pushed the boundary of using quantitative EEG in the sense that we incorporate what's known as the cross spectrum, which means that it is all functional connectivity in spectrum as a mediator. And we've shown that it's a mediator between malnutrition and cognitive impairment at school age. And these are tools, let's say, not only having data that's known, but also now to be able to use it to explore mediation so that it can be used in clinical trials, also in study of what is the effect on the brain of different factors. We've also carried this out for COVID-induced brain disorders. This is a call in which Muming Pu from China, Alan Evans, and Mitchell Valdes from the Cuban Center for Neuroscience, we make a call to study long COVID, but not only due to infection of the brain, but also to social isolation. And we have studied that even social isolation can carry out modifications of brain functional connectivity as regards to the normal population, but the changes are more severe in people who did have the infection. So this illustrates our approach to trying to look to solutions for huge, let's say, health problems using electrophysiological techniques. Our most recent publication is highlighted in the International Journal of Biomedical and Health Informatics, and this is recovering work. We now know about FUF. We know about aperiodic components and rhythmic components. This was already described by Roberto Pascual in the Cuban Center for Neuroscience in 1987. And we now have a method that was highlighted, as I say, in this journal, which substantially outperforms other methods. And it essentially decomposes the EEG spectrum, not using parametric functions, but non-parametric functions. Therefore, it is very flexible, and that gives it the advantage. So This is just to illustrate the methodological improvements that will be coming and be incorporated soon into EEGNet. So having given an overview of why we're doing this, what are some of the results that we have, I now can turn over to the basis of this, which is going to be EEGNet, the platform for sharing the data, but also for sharing the algorithms worldwide. So I think Christine is on now.
Thank you so much, Pedro. I'll just share my slides. And can everyone now see my screen?
Yes.
Great. I will continue. I'll continue from Pedro's remarks. Thank you very much. So what I'm going to do today is present an overview of the EEGNet data and analytics platform, which is found at eegnet.loris.ca. I'm also going to say thank you very much to those of us to our supporters who were able to make it today. This includes the Ludmer Center. This also includes, as you heard, Sabina Antonescu from Brain Canada. And this project is made possible with the support of many collaborations, including the Global Brain Consortium, and additionally some funding from Canary, in addition to our partner institutions, Brock and Laval, and the Canadian Open Neuroscience Platform. I'm just going to move the screen forward. So today I'm going to speak briefly about the platform context and technical objectives as we move forward with this grant. We'll have a brief video demonstration and also provide information just after that, additional details on how to request an account at eegnet.loris.ca, since we would definitely encourage you to take time today, if you haven't already, to please do sign up for the platform and get involved and let us know your feedback. And I'll go over a few ways in which you can get involved further. So with that in mind, going back to our grant start in February 2021 and the objectives that Alan outlined, many of our deliverables have in fact been delivered in the past 3 years, and we are still working on adding additional validation toolkits for standardization of EEG BIDS data. We're always welcoming additional data sets onto the EEGNet Hub, and we're also still developing ways to integrate analytics and to sync the metadata and the results of those back to the EEGNet Data Hub. As Alan mentioned, the goal of the grant is to provide a platform that is an open data sharing and analytics hub, currently connecting over 50 researchers across Canada with an even larger network of international partners, in partnership, of course, with the Global Brain Consortium, in order to build a truly collaborative EEG platform for advancing neuroscience. The ways in which this is designed to operate is that EEGNet brings together standardized data formats with annotation workflows and open source analysis tools to enable that cross collaboration, resulting in translational applications for the early detection of biomarkers and for translation in machine learning and applications in global health. To that end, EEGNet leverages the community-driven standards emerging in electrophysiology across a network of clinical, technical, and scientific researchers and advisors to collaboratively build and to validate these open EEG BIDS annotated data sets. And this is where we particularly look to increase community engagement in the annotation and validation process. So the Open EEGNet Collaboration Hub also unites head tag annotation workflows with open source analytic toolkits containerized for deployment on high performance computing infrastructure as part of this technical platform. As Alan mentioned, the platforms are a combination of the open source Loris Data Hub and the open source CBrain Analytics Hub. This is infrastructure developed at the McGill Center for Integrative Neuroscience at the Montreal Neurological Institute and Hospital and supported across Canada. The interchange between Loris and CBrain is what allows the Data Hub and the Analytics Hub to work together. As a result, we have an integrated open hub for building these analysis-ready and validated EEG BIDS standardized data sets. So that brings together the data platform, the online workflows, the data sharing, and the analytics in order to build these resources and these research assets that are ready for applications in machine learning, biomarker detection, and global health. Since the start of the grant, we had a few delays due to COVID and staffing challenges in times where it was nearly impossible for some groups to collect data. So we've been very mindful of some of the longer timelines needed to execute on some of these deliverables. Starting 2.5 years ago, we piloted a data platform to support EDF and BID standardized data, and that's compliant with the brain imaging data standard. We followed that up with the deployment of the data hub, online workflows, data sharing, and analysis hub with a few initial tools, including the harmonized EEG toolkit, which you'll hear about later from Jorge-Bosch Bayard, and the brainstorm tool released from our colleagues at Laval, CERVO, which Philippe Albouy will address shortly. In fall 2023, we had our platform launch. If you missed that, the entire video is viewable on YouTube, as this will be after. So we invite you to check that out. At that time, we released more data sets and tools linked with the C-Brain processing platform. And through this spring, we've added online workflows for signal annotation and head endorsement. So some of our data sets and tools have grown over this time, as Alan mentioned. We're also looking to grow our user community of those logging onto the platform. And we are also growing this year, especially the number of conference talks, presentations, and papers, as Alan mentioned. In addition to this, if you log on to eegnet.org, our public-facing website, you'll see additional community tools and data sets listed. These are research assets that the community has let us know about, and increasingly in 2024, we'll be cross-linking these with the Canadian Open Neuroscience Platform. So as I mentioned before, the priorities for platform progress in 2024 and going forward include community engagement, where we're looking to you, the research community, to get engaged in annotation of data and endorsement of head tags in order to build that validated data set that serves as a foundation for machine learning and other applications. We're, of course, always looking for contributions of open data sets and analytics tools. And we're increasing the outreach with analytics workshops and technical guides. In addition, there are presentations and onboarding workshops, as Alan mentioned, planned at OHBM this summer in a pre-conference course, at the MOBI workshop shortly, led by Scott McCaig, and at the Clinical Neurophysiologist Conference in September. Some of our ongoing technical objectives as we move forward through the 2024 year are to increase the support for channel-based events and annotation, curation of events from broader tag sets, and versioning tags and dataset tracking, in addition to contributor roles and granular permissions there, and increasing automation of the mechanisms for contributing datasets. So with that, I'll give a preview of the platform. We're going to play a video, two video clips shortly. These are taken from the videos already on the YouTube channel of the Global Brain Consortium. So we certainly invite you to check that out. And I'll roll the clip now.
To request access to EEGNet, click the link to request an account and register to receive an email with your login credentials. Remember to review and accept the terms of use before you log into EEGNet. The dashboard summarizes the data sharing and analytics resources and workflows available through the EEGNet platform hosted on LORIS, an open source database built at the McGill Center for Integrative Neuroscience. Recent activity on EEGNet analytics tools mounted on the C-Brain distributed processing portal are displayed in the top right corner. A visual breakdown of the datasets or projects gives different views of the composition and size of data available on the EEGNet platform. Click on the label of a dataset to learn more in the project information panel below, including publications, how to cite this dataset, and task descriptions. To view the EEG signal data, click through or use the menu bar to access the electrophysiology browser to view the session recordings from any project dataset, raw or derivative. All available recordings can be reviewed in the electrophysiology browser module. Search for the recordings of interest and select the dataset type in the linked column, raw, derivative, or both. for a single session or visit, scroll down to view the signal plot and its interactive features, metadata, and a basic electrode map in 2D or 3D view. All downloadable files are available under the signal plot. When viewing one EEGNet recording at a time, you can use the interactive signal plot to review and edit recording events and tag specific events for all channels of a recording with specific head or score tags. Click Show Events panel to view all the events in the current viewing window. Press the I button to hide and show events. The Tag button shows any head tags associated with this particular event. Press the third button to edit the event. Hover over head tags to view additional information. You can add and remove head tags already used on this dataset and score artifact tags with this menu.
Thank you. So that is a clip narrated by our lead developer, Jefferson.
To request
Jefferson Casimir, who's here on the call with the rest of the EEGNet team. That clip is from our YouTube Global Brain Consortium channel video of the platform demonstration from last November, and we'll be updating that video shortly for this spring. We certainly invite you to check out the rest of those videos, including the presentations from our launch last fall, for more context, for more information, for a sense of the platform, and of course, to show you how to navigate the platform should you wish to request an account following these steps here. So we do recommend Chrome, and you'll be asked to review and accept the terms of use, which are part of the Canadian Open Neuroscience Platform Ethics Framework. You'll be asked to reset your password after it's sent to you by email. And we would also invite you to request a CBrain account so that you can access the EEGNet tools and processing portal. So that's how to get started basically with the platform. And moving on from this, if you'd like to get started to get engaged with contributing dataset, tools, and annotation on the EEGNet platform, you could scan this QR code or just reach out to us on our website to fill out some basic information about things you'd like to share and to get in touch. You can also select a few options based on how you'd like to share your data set or tool. I'll be walking through in the next slides what you might consider in terms of what you can do and what you might want to do with EEGNet, as well as the Canadian Open Neuroscience Platform. And then the third step is, of course, to prepare your data or your tools and to send it to us directly. Just to note that for contribution to this platform, citation is something that we ask everyone to provide links to and information for in their licensing. And in that sense, it is citation of your research assets is required. Authorship's not guaranteed, and we can't technically enforce citation, as is the norm in academia. But to move on, in terms of how you would share data with EEGNet for the purposes of visualization, annotation, and analytics, there's a few things to know about the steps towards this. So the first thing you'll want to do is look at your consent and your ethics framework to discover what you can do with your data. And a fantastic resource for navigating this is the Canadian Open Neuroscience Platform Ethics Toolkit, which is a framework governing all of EEGNet as well. And the second point is to look at what you would like to do with your data. So for example, if you'd like your data to be downloadable, everybody knows there are many ways in which to make that available in our open ecosystem across neuroscience. But if you're interested as well in having your data be visualizable, queryable across in an integrated platform with other data sets to enable quality review, to enable annotation, and to contribute it to these gold standard global data sets, that's where we would invite you to really look at EEGNet for that option. Multimodal databasing is also an option as we support MRI and other modalities as well. So moving forward from there, EEGNet allows researchers to visualize their data on the platform in addition to annotating it with head tags, such as the hierarchical event descriptor tags, also enabling community annotations for collaboration on your data set, and analysis of this data with tools on CBrain on the high performance computing clusters that we have as part of the infrastructure. Canadian Open Neuroscience Platform also allows distribution and sharing of data. This is more commonly done by linking a Zenodo or an OSF entry on the CONP portal, which we invite you to check out. In addition, CONP can host data sets on their server if they require, if it's a very large data set, for example, that requires a physical site that's protected and custom metadata. And the option to link your data to the CONP portal through DataLad is always available. So the third step would be to get in touch and to prepare your data and then send it to us. So for that, you can reach out to us at our contact information below. And we always have open office hours on Zoom every Tuesday at 2 p.m. Eastern. Feel free to drop by on Zoom and get to know the team. Common tools for preparing your data and sending it to us that we make available and that we'll be very happy to walk you through include converters, such as the EEG to BIDS Wizard standalone app that helps you standardize your data and convert it to the brain imaging data standard. There are EEG uploader tools and dedicated SFTP transfer processes. We also offer templates for standardizing metadata and containerizing tools. And there are also boutiques descriptor templates to enable containerization of tools on CBrain to facilitate the process and to lower the technical barriers or the sophisticated informatics knowledge that one might expect from certain of these platforms. All of these tools are there to make the process easy, straightforward, and to facilitate this to use your time well as well. So moving forward from this, I'd just like to thank the team involved, including all of our global advisors and members of our scientific and clinical and technical advisory boards, including members of the Global Brain Consortium for all of their contributions to the design and the conception and the guidance for this platform. The EEG networking group is here on the call today, so thanks to all of them for their efforts, and additional support from other developers within the McGill Center for Integrative Neuroscience who contributed over the years to much of this infrastructure, and we certainly would like to recognize their efforts. So with that, I'll close my remarks and say thank you very much for joining us, and please don't hesitate to get in touch. Thank you. So over to our next speaker is going to be Jorge-Bosch Bayard.
Okay, hello. Thank you everybody for joining this colloquium, and thank you for allowing me to make this presentation here. I am going to briefly introduce two methods for quantitative EEG analysis that we have developed and are already incorporated into CBrain. I think the intention of introducing EEG-based methods in CBrain is a long goal that Dr. Pedro Valdes and Dr. Alan Evans has had for many years. But I think a very important step was the creation of the Cuba, Canada, China axis, where they made the goal of creating a neuroinformatics platform for developing methods based on EEG integrated with all the tools developed at the Montreal Neurological Institute, and including these methods in CBrain as analytical tools. Other two key moments for this, I think, were the creation of the Global Brain Consortium and also the EEGNet initiative that brought together other groups, many groups working in the EEG research, who has also contributed to the development of analytical tools to be incorporated freely in CBrain. I just want to mention two of them that have been already mentioned by Christine. The pilot-led pipeline developed by Broad University, and the brainstorm tool developed at Laval University that I think will be presented here too. In my case, I'm going to just present two quantitative EEG analysis that we have developed. The first one is the tomographic quantitative EEG analysis that have this name tomographic because it was the first tool that we developed for quantitative EEG analysis at the sources, and the newer version of this analysis that is the multinational harmonized quantitative EEG analysis. The QEEG is the traditional quantitative EEG tool based on the transformation, taking the signal at the time domain, converting to the frequency domain by means of the Fourier transform, and taking the logarithms of the spectra as descriptive parameter to characterize the EEG activity present in some EEG recording. Then from this descriptive parameter and the recording of a normal database, obtain the typical measurement of this descriptive parameter in a healthy population. And with this thing, it makes possible to characterize deviation from normality in patients by calculating the T-transform probabilistic normalized measurement that allows to know how different from the normality is one person that is a new person that is measured. Just to highlight that the new harmonized multinational analysis or methodology developed is just a continuation of the work that we did in the early 90s. I have put both of them in the same diagram. Both the harmonized, multinational harmonized QEEG analysis also follow the same steps, the same flow of diagram flow, incorporating new techniques and modern techniques. One of the most important was the inclusion of the batch norm harmonization that already Pedro mentioned. This batch harmonization is an algorithm that allows to harmonize EEGs coming from different EEG machines to make them comparable. This allowed us to create for this paper, for this work, a multinational database collected in more than 14 sites in 9 countries, coming from different equipments, harmonizing all of them and putting all together more than 1,600 soldiers from 5 to 95 years old in a multinational database that, from which we obtain the technology. The other key extension that we had with this harmonized multinational database methodology was the inclusion for the first time not only the log spectra of the EEG, but the full cross-spectral matrices. It means that the cross covariance between electrodes, which added a new more information to the QEEG analysis than what we had before. As some products of this, we also published the 2 databases, normative databases collected in Cuba from normative population around the 90 years, and also the one that Pedro already mentioned, from the Cuban human brain mapping that also contains EEG and contains a MRI signal together with some psychological tests. The QEEG is already incorporating in C-brain. It has this interface, and from calculating the QEEG, where you can obtain the raw and the normative spectra, and you can see the graph, you can see the activity, raw and normal, or fixed scores, both at the sources and at the scale, both for the narrow band model and the broadband, the measurement of the broadband models. In terms of the harmonized multinational QEEG, also is incorporated to C-brain. It allows different EEG formats. So it allowed to get 3 EEG from the bits format, from the EDF, PDF, PLD, and the set created by the EEG lab, as well as a test format. So which are the most popular, most common format used nowadays in EEG, and it creates the QEEG derivatives that are also stored in the EEG mix format using the HDF format, which is a very compact and efficient way of storing complex data like this. Why do we need we need the new harmonized QEEG? What is the contribution of this harmonized QEEG? Besides the batch harmonization that allows comparison with different equipments, the inclusion of the cross-spectral data and descriptive parameters allow to improve the classification power of the QEEG very much. So it's a very great improvement with this. This is an example of where we are doing, in which we are analyzing 3 groups of dyslexic people against a control group. I don't want to describe the groups, just to mention that if we look, for example, don't pay attention to so many colors, just let's concentrate in this yellow. What we are looking here is the scores of the classification, biomarker classification within of these 4 groups. So if we look at this, we have here the scores of the classification for the group of the control group using the QEEG technology that only includes the logarithm of the spectrum. We obtain a very good classification of the control, the 3 dyslexic group, but there is not any separation of the 3 dyslexic group. You can see the 3 yellow bars here. The classification from the dysagenetic and dysphonetic is 0.58, 0.64, 0.65. So the classification, it is the area under the ROC. This is just under the random level. However, when we use the complete, the full cross-spectral matrix, for example, let's see the blue color, the control group. So the biomarker procedure find biomarkers that allow differentiation of the control group, very full differentiation, and complete differentiation from the 3 dyslexic groups. And you can see here that there is a big differentiation also between the 3 dyslexic groups. So the 3 dyslexic group that were not able to be separated by the log spectrum, when you include the correlation between the different electrodes, then it is possible to pass from 0.58 to 0.81 to 0.94 and to 0.78. So it's a very dramatic improvement in the classification power of the EEG using the new descriptive parameter added by the harmonized QEEG methodology. The method that we use for the biomarker selection in the figure that I presented before is another contribution. It's a statistical methodology that uses different levels of cross-validation and provides allows to obtain a very stable set of biomarkers, sparse set of biomarkers, very stable. In the case, especially small sample. So this method is based in the random repetition for cross-validation in the 2 steps, and then it allows first to obtain stable classifiers, and second to give very robust estimation of the ROC incorporated to CBrain, just that it is still in the test server. It's not, has not been put in the official CBrain, but this is also another method that can be very interesting for people doing research and wanting to obtain stable biomarkers. Then there are other contributions for in terms of this. It remains still at the level of the scalp. We didn't develop it for the sources. This is a work that is being doing by Ronaldo Garcia at Pedro's lab in Chengdu. He is developing another method that allows to go to the sources, and also that solve a drawback of the previous method, because the harmonized QEEG as it is now, it works in it doesn't work in the original space. It makes a transformation of the cross-spectral matrix to a tangent space, and it means that we lose the when we talk about the biomarker, we took the anatomical position of the electrodes, so we cannot talk about relation between electrodes directly, because what we have in the transform space is a combination of different electrodes. But the method that Ronaldo is developing works in the original space. He said will be able to give direct interpretation of the results in terms of the topography, and also will be able to use in the sources. And then I think this will be the next step to put in the CBrain. And other tool that has already been published and is a very complete toolbox for the neuroinformatics platform that integrates almost all the steps, both for using the QEEG at the level of brain templates or using individual MRIs. Has been already finished and published by Arioski, also from the group of Pedro Valdes. By the way, he will defend his PhD thesis with this software next week, and I think it will be also another very soon incorporated to the CBrain tool. I think these are some of the toolboxes that we already have developed and will be able to be incorporated to continue increasing the power of analytical tools in CBrain. Thank you very much. Then I pass the floor to the next.
Thank you very much. Next, we'll have Sid Segalowitz from Brock.
Hi, I'm just going to get my screen started.
Great.
Here we go. Okay, I'm going to talk about some of our studies on attention and attention control in younger and older participants, and give some examples of the data sets that we've either uploaded or will be uploading soon. As you'll see, they do not need to be huge, as the point is to accumulate the data across labs. I should just mention at the start that all our data involve 128 channels. This, of course, makes many of the analyses much more efficient. So the data I'm going to talk about so far come from two of our labs. I'll be giving brief descriptions of our research goals so you can see how they vary considerably. Both are associated with lifespan. Our labs are associated with the Lifespan Development Research Institute at Brock, and I'd like to acknowledge the help we've had from them. From our developmental neuroscience lab, the group interested in child and adolescent mental health involves myself and Professors Ida Tikakilik and Aaron Panda, both from the Child and Youth Studies Department. And from the second lab is the Neurocognitive Aging Lab, headed up by Karen Campbell, who's our CRC in Cognitive Neuroscience of Aging. And the data I'm going to talk about were collected by her PhD student, who's about to finish soon, Sarah Henderson. At the Developmental Neuroscience Lab, we have 3 projects I want to mention. First one is in collaboration with Pasto Mental Health, which is our local center for mental health in children. And we're focusing, as you can see, from age 8 to 17 years on ADHD and anxiety issues. We have so far about 100 participants with psychological needs, some with psychological needs, and of course, healthy control volunteers as well. And we're focusing on issues related to, as indicated here, ADHD and anxiety disorders, as these are by far the most prevalent reasons for people contacting the clinic. The EEG battery it has resting EEG, eyes open, eyes closed, visual oddball tasks with a variety of conditions, and such. So these are very standard EEG measures. We're interested in ERPs, but as you see, also in spectral analyses. The data are being examined now. We don't have any longitudinal results to tell you about yet, but we are hoping to be able to collect such data and then follow up later as children go through their clinical treatments to then evaluate whether the EEG that we collect early on is associated with success or non-success with the treatment. In other words, are we going to be able to use them to help predict success likelihood? The data we're uploading at this point are basic demographics along with the EEG to EEGNet. The second data set was collected as a master's thesis by Carolyn Hare, who's finishing her PhD at Western University now and is soon to start a postdoc position with Sarah Lippé at the Université de Montréal, who we hope to hear from later. Her study involved 60 non-clinical university students on dimensional measures of ADHD and inhibitory control, currently in review at psychophysiology. Hopefully we'll have it out soon. The EEG data with basic demographics are being loaded up to EEGNet shortly. For the EEG inhibitory control measure, we're using the very popular frontal central N2 component in response to response inhibition in a go-no-go task. Also in comparing with trials with distractor and non-distractors. So it's a fairly complicated paradigm, but of course it'll all be described. What we found was that trait anxiety moderated the ADHD trait scores in predicting the N2 amplitude. In other words, ADHD trait scores did not predict N2 amplitude alone, but they were moderated by trait anxiety. And so rather than being just highly comorbid, they're not additive, but interactive. That's what we've concluded. The third set of data that from our developmental lab that I want to mention involves the aperiodic EEG spectrum and its ability to identify ADHD. Pedro described it very briefly early on, the wonderful, wonderfully named algorithm, FOOF, which basically gets at the slope of the spectrum. We all know that as you go to higher frequencies, the amplitude, the power drops, and that slope has turned out to be of interest. A quick description for you who's not familiar with it. It was first, as far as I know, it was very early on described as important in evaluating how we measure, for example, alpha in EEG, because as you can see, as an example here from Robertson et al.'s 2019 paper, nice description, the same level of power of, for example, the alpha peak can be interpreted rather differently depending on what the rest of the spectrum is doing. And therefore there's a confound there. that can be interpreted in interesting ways. I'm not going to go into that now, obviously. But there's that confound we want to pull apart. And what Robertson and company have shown, and others have as well, is that children with ADHD, these are young children with ADHD, if they're unmedicated, show a higher slope value, which is essentially 1 over the frequency ratio, in accounting for, or at least relating to, the ADHD. Generally, the EEG slope is greater, and in this figure here, it's showing that at all the electrodes in the ADHD. But normative studies also show an age effect as well. So in order to examine this before we get to our Pathstone data, we wanted to see if we could build a model of how ADHD and age, and for that matter, sex, interact in predicting ADHD. And here's an example of using an open EEG database. We got data from the Child Mind Institute's Healthy Brain Network that Alan mentioned earlier. And what we found from their database, which is a large one, and it's the sort of thing we're hoping to, of course, build for within EEGNet as well. 1,200 boys, 500 girls with ADHD, and then the brain controls. That there's some interesting interactions. There certainly is an age effect. As age increases, the slope of the spectrum changes and goes from higher to lower slope. And there's an ADHD effect. And that will confirm what Robertson and others have reported. But on top of that, there's some differences between boys and girls. So for example, when you compare control boys and control girls, there's a crossover effect because the control girls are showing fairly flat slope with respect to age changes. These are cross-sectional. With a crossover at about age 11. The ADHD have an earlier crossover point, but notice, of course, that they're steeper. And so this will give us a heads up on what we are going to expect to find with our clinical group that we're testing. So in other words, the relationship of the aperiodic EEG and ADHD depends on both age, sex, suggesting a much more complex relationship than we've seen in literature so far. The data from Karen Campbell's lab
I think we might be having some internet
data sets I'm going to talk about, a different kind of questioning. This is the normative. Sorry, can you hear me all right?
I think you might have just cut out just a moment. If you'd like to.
Oh yeah, I got a note saying it's on my internet's unstable, so I will talk a little slower. That may help.
It's great now.
Okay, great. The first one's a mind-wandering study. It's recently accepted for publication in Journal of Cognitive Neuroscience. There's the listing up there. What they were looking at was in 49 young adults, 40 older adults, with eyes open, eyes closed, resting EEG. But in addition, the SART task, the sustained attention to response task, which you probably may be familiar with, where the participant, it's like a go, no go, but it's mainly go. You press the key almost all the time and occasionally have to inhibit it. And they give it for 25 minutes, which is a bit challenging. And of course, people start to lose concentration, and then the mind starts to wander, which is what they're looking for. They would look for changes in the P1 component and the P3 amplitudes to the stimuli. With respect to mind wandering, they measured the mind wandering by looking at the increase in variability of the reaction time. So when you were off task, of course, the reaction time varied considerably. They found that the P3 amplitude reduced went off task more in the younger group. But there was no change in the P1. So they argued that mind wandering affected stimulus at the evaluation stage, but not the perceptual stage. My main point, of course, is in presenting this is kind of data that other people may want to make use of for other purposes. Their second study, involving 31 young adults and 35 older adults, if you may have noticed when I think it was Christine was presenting the data that is available on EEGNet, these are already uploaded, and it's called the social sound localization study, which is part of that was the cover story for for what the participants were doing, but really was a memory study from their point of view. And in this study, they were looking at what happens when they present sounds, environmental sounds, everyday sounds, to the left or right speaker, half of which were paired with a visual image. There's 30 minutes of this data collection. At the EEG session, the sounds were presented, and for half of the subjects, they were asked to try to remember the location it was played at earlier. For the other half, they also were asked to try to remember the picture. So there was presumably an implicit memory of the picture sometimes, non-forced, and the other was voluntary remembering. They found that the older adults showed pronounced alpha event-related desynchronization, or ERD, regardless of the intention. They also showed greater ERD for paired than unpaired sounds at occipital sites, which the young people did not, suggesting that all of this, that the involuntary memory is largely preserved with age, and that was the cognitive question they were looking for. So what I've tried to describe very briefly is just the variety of paradigms that are possible, but of course the data can be used for many purposes. And these are just some examples of what we're uploading. So I think that's it. Thank you.
Thanks, Ed. Philippe Albouy will be presenting next from Laval, CERVO. Great, we see your screen.
Do you see my screen?
Yeah.
Good. So thanks, I'm happy to present what we are doing at CERVO in the context of EEGNet. So I want today to present the analysis tool that are available on EEGNet, so mainly with two aspects. So EEG data analysis with Brainstorm, and what we are developing now, that it's more ongoing project, is trying to incorporate machine learning tools with scikit-learn. So I did this presentation with Corentin Labelle, who is the software developer here at CERVO, and I do this presentation on behalf of the EEGNet team at CERVO and Université Laval. So the team is with Isabelle Blanchette, Charles Fecteau, and Céline Bastien. And all this work has been done in collaboration with the Brainstorm project. So you might have seen this slide before in the two previous talks. So Christine and also Alan did a really great description of the entire workflow, combining Loris Data Hub and CBrain Analytics Hub in the context of EEGNet. And what I want to do today is to present a bit more just examples or more details for the CBrain Analytic Hubs, with the specific example of Brainstorm and what we want to do also with the scikit-learn functions. So to do that, why have we chosen Brainstorm as a first tool to try to incorporate to EEGNet? And I'll try to explain and develop why this tool is efficient. And all the slides I will show here are from Sylvain Bayard, with the head developer of Brainstorm. So when we want to do EEG analysis, we need to find a good software with that will allow us to have good scientific practices. This way, we have tried to implement and put Brainstorm on CBrain, because we consider that this tool has all the relevant tools for EEG analysis. It's easy to learn, easy to use. There's a really big community that is welcoming and supportive for the the using of this tool. The tool is also evolutive, interoperable, and enables shareable processes. The main problem about this tool was, and I will insist on that just after, is that it's developed with MATLAB, and not all labs have access to MATLAB. So what we want to do, and what I will really present today, is trying to make this tool available without MATLAB via CBrain and EEGNet. So we really want something that can be easy to use and free to the users without MATLAB and computing all your analysis on CBrain. So just a history about Brainstorm. So it has been developed, so the leader is Sylvain Bayard at McGill University with his colleagues. They have started working on Brainstorm in the 90s, and now it's a quite well established toolbox and software that has many users, so more than 40,000 users. Many training sessions have been done, so this software is more and more used, more and more cited. And the main important point is that this software contains a lot of tools that will allow you to do all your pre-processing and classic processing steps for EEG, but also for MEG, for intracranial EEG, for FNIS also. And plus, it combines different tools from other software, other toolboxes that I will present just after. So the idea is that when you do and you want to do an EEG analysis, there's many different processing steps that should be considered. And Brainstorm, using the functions developed in Brainstorm, but also many functions from other and many tools from other toolboxes, allow to do all these different steps. So we start with data collection, so data review quality. So this you can do it via EEGNet visualization toolbox, but you can also do it via the interface with Brainstorm. Then for all EEG data, usually we do artifact detection and correction. For that, we can use different software such as SSP, signal space projection developed for MEG in Brainstorm, but also ICA in EEG Lab. And these functions from EEG Lab are also incorporated in Brainstorm. Then you're going to do, if you are interested in cognitive neuroscience and you have different conditions, you can do event registration. Then you will be interested in doing source modeling. To do that, you can compute your noise modeling, but also your head modeling. And for that, we also have access to different functions from different groups. It can be the overlapping spheres developed in Brainstorm, but can be also the OpenMeg function for realistic head models. Then usually when we are doing EEG analysis like this, we try to do source modeling. And here again, in the community, there's many different tools that are available. And those tools, most of those tools are already implemented in Brainstorm, such as dipole fitting that has been developed in Philippe, can be also beamformer of a Philippe function, minimum norm developed in Brainstorm. Then once your source imaging is done, you can do your signal extraction with classic analysis, so event-related response, state response, ongoing activity for resting state or sleep. Then different measures can be extracted in the time domain, so latency amplitude for the event-related responses, or more spectral and time frequency composition. Here again, you can use very different approaches, such as FFT, power spectrum density, Hilbert transform, Morley wavelets, Philippe multi-taper, are also implemented in these tools. Other metrics that are a bit more complex but still implemented are cross-frequency analysis that are well documented and well developed in this tool. And then you can do also functional and directed connectivity between different regions, or end-by-end connectivity if you want. For the functional connectivity, many tools are available, like coherence, phase locking value. Directed connectivity can be estimated with Granger causality, or even with the phase transfer entropy. Then once you have your metrics of interest, you will do your statistical inference that are implemented in Brainstorm with parametric test, non-parametric test, and again, Philippe function for cluster-based permutation testing. So based on that, we were considering that many of the relevant tools are present in this toolbox. That's why we're thinking, okay, could we try to make it available to the community, but without MATLAB? So then new features also that are developed in Brainstorm, they are trying to develop a lot the analysis of stereo electroencephalography or ECOG. So the software is still continuing to be developed. One good aspect of Brainstorm is also the documentation. So the website is well documented with a really large community like this, more than 4,000 users. Everything is quite well described how to install it or to do the different pipelines, and the tutorials are really well described. So we consider based on that the tool is easy to learn. But another thing is that if we want people to use it, the logic of Brainstorm at the beginning was you should be able to do your data analysis without coding or without scripting. So you can perform all your analysis only with the GUI if you want. So it's easy to use, easy to learn, and if you have problems for a given step in your analysis, you can still discuss with the forum on Brainstorm because the community is quite supportive. You can see that every month there's around 800 posts that are done on the Brainstorm forum. So many tools are available with Brainstorm, and is also quite foolproof and stable. So there's many ways of identifying the artifacts, checking the effects of your different processing steps. The community is also large, so it means that the software is evolutive, and many new updates and addition of new tools from different softwares. So I was mentioning a lot field trip functions before, are added continuously in Brainstorm. And that's where I think that's one good aspect for the EEGNet is that the software is interoperable. So for doing all the different processing steps I was describing before, you need with the EEG to play first with the anatomy. So you have access to free software functions, Brain Visa, SPM tools, and many of the different steps I was showing before are also developed by the other softwares like ICN EEG Lab, source reconstruction, so Beamformer, cluster-based statistics with field trip, dipole fitting, multi-taper in field trip, decoding analysis with Emily Python, anatomical analysis with CAT 12, etc., etc. So the toolbox contains many external hubs that are analytics that are usable in only one software. And one interesting aspect is that when you have determined your pipeline, then you can extract the different process to share it with the community. But today, the main problem of Brainstorm and other tools is that MATLAB is not free. It's true that there's a compiled version of Brainstorm that can run on all operating systems. But if you want to analyze 1,000 participants, you cannot do it manually with the GUI as a repetition like this. So that's why EEGNet is trying then to help the community here by giving a tool that will allow you to run all your Brainstorm pipelines on CBrain. And I want to show you how you can do that step by step. And the idea was to have something really easy to use and with only 2 files that you're going to upload to CBrain. So I want to show you how you can do that. So if you go to the main page of EEGNet, there's a list of the different tools. And then the idea is just going simply to the GitHub link here to see how you can compute and process your different analysis on CBrain using Brainstorm. So when you click on this link, you will open this page on the Brainstorm tool that describes that this tool will allow you to run Brainstorm functions on CBrain. And there's tutorials how first to create and use the CBrain tools that you can use. The idea is really simple, is when you want to do an analysis on a given data set, you just have to create a pipeline. And the pipeline can be saved in just 2 different formats, a .mat that is extracted from Brainstorm, or you can do it just writing a JSON file. So we'll first show the mat format because it's the easiest way of doing it. If you don't have MATLAB, so imagine you have 1,000 participants on EEGNet, so on LORIS, you want to apply the same analysis on all subjects. Then you can just download one subject locally on your computer. Then you can go to the brainstorm page and download the compiled version of brainstorm, meaning that you don't need to have MATLAB. Then with this example subject, you will define the parameters for all your processing steps. So you want to do, for example, here bandpass, you want to detect the error bits, you want to do an ICA, etc., etc. It can be done for all the functions and all the processing steps that are available on brainstorm. Once your pipeline is defined, so here is just defining with an interface the different parameters you want to use for your analysis. Then you can simply save it as a MAT file. Once you have this MAT file, you go back to CBrain. The other option, if you don't want to use the compiled version of Brainstorm, is going to the GitHub again and see all the different process that can be run in the format of the JSON file. And you have a clear description of all the process, what they are doing, and what are the parameters that you need to do. So if you want to do the same process as before, so featuring detect error bits, ICA, time frequency analysis, you can just go to this website, take the parameters you need, put everything in your JSON file, following the tutorials available on the GitHub, and then save your JSON file. Once you have that, so you have two options, the MAT file extracted from the compiled version of Brainstorm or the JSON file. Then you can go to CBrain. So there's many tutorials and guidelines to create an account and download files, upload files in CBrain. So the idea is just to create a project, add your data, and then go to your project and select the data sets. So usually it's going to be in the bits format on which you want to apply your pipeline. Then the process is really easy. You have only two files. The pipeline has been defined on an example subject, extracted, then to a MAT or JSON file. You upload that and your data set to CBrain. You select your files and you go to the launch Brainstorm task. You select your version, you select the different parameters you want, where you want to compute it, etc., etc. But the two important fields are the bits data set you want to analyze and the pipeline. You select only those two aspects, those two components, and you're going to run your analysis. Then the output will be derivative. So in the bits format, your process data that has been applied to all the subjects you have selected here. So this way you can use the GitHub, not the GitHub, sorry, you can use the interface to create the different pipelines and the different process you want to run. And then you just have to apply those pipelines to all the participants in your study. And you can do that for all the different steps, all the different analysis I was presenting before. What I want to, just to conclude, we are trying now to do that the same principle for machine learning tools. So we are just starting, we started a few months ago. So we are trying to put scikit-learn tools in CBrain to apply it to the data that is already present in EEGNet or use different data sets. And the logic we want to apply is a bit similar to the one we have for Brainstorm. The idea is to have a given data set and then apply, so create a process, a pipeline that will allow you to do the different machine learning analysis on your data set, upload it in CBrain and just run the analysis. For now, we are trying that with simple supervised learning models, so ridge regression, SVM, perceptron, or logistic regression, just to see how we can implement all of that together and make it really easy to use for the EEGNet users. The logic will be similar. We will put the data set we want to analyze and then the pipeline for the machine learning to then get the output of the classification. So just to conclude, in this general topic, so we will try to add more and more tools on CBrain, trying to combine the tools that are developed in the community here to help the researchers who do not have access to MATLAB or need really high performance computing cluster to do their analysis. So I want to thank you for your attention, and I want to have a special thank you to Corentin Labelle, who is an EEG developer in our lab and who is making all of this possible. Thank you.
Thanks very much, Philippe. And thank you, Corentin, too. With that, we'll move on to our speaker, Sarah Lippé from Université de Montréal.
Hello, everybody. So wait a second, I'll just oh, this is okay. So does that work for you? Oh, no.
We see you on the screen. For the screen sharing, I think that might take just one more moment. Would you like to try again? There we go.
You see it? Okay.
We see
Okay.
your presenter view instead of the
Oh.
just the slide. Perhaps you might like to switch screen on Zoom. Or...
Maybe I just do that. Okay. Yeah, sorry about that.
There we go. Perfect.
Yeah. Okay.
Thank you.
Sorry for the delay. Yes, I will switch gear a little bit here. I won't be presenting tools. I will be presenting basically what my team and I are doing, and really maybe highlighting how EEG can be highly useful in neurodevelopmental disorders, more specifically. So I'm Sarah Lippé from University of Montreal. I'm a neuropsychologist by training, and I digged into EEG during my PhD. So it's a long time ago now, more than, I think, 25 years. And I decided that it was a great tool to assess brain maturation, brain development abnormalities because of its potential, as you all know. So my team and I are extremely interested in trying to understand the consequences of several risk factors for neurodevelopmental disorders, but today I'll focus a bit more on the genetic risk factors. I would like to highlight that it's the 100th year of EEG this year. It might have been said several times, and I think we made great progress since 100 years, but maybe not enough, and probably having the community together will accelerate these advancements. So what we typically do with the EEG signal to assess neurodevelopment is to extract different features from the signal. And there's a lot of those signals, and those are sensitive to brain maturation. And these features kind of develop in a high speed. What I mean by this is that we have new features that seem relevant very frequently, and we're all very passionate about trying to understand what they actually refer to in terms of brain disorders, brain development, and aging. So what EEG is sensitive to is really a number of factors, and this is lucky for us as neuroscientists and neuropsychologists. But it's also extremely sensitive to age variations. So what you see here is a set of nearly 250 participants where we assess the effect of age on the cross correlation of theta and gamma coupling. So this is major as an effect, as you see. Of course, there's also other aspects that are that influence this correlation, including a genetic mutation, including IQ, including sex. So luckily for me, you can see here that this influence of age is really high during the first 10 to 20 years. And it's also very influenced by aging, but there's a moment of latency, I would say between 20 to 30 years of age. The other aspect that is interesting to see in infant brain is that it's very sensitive to age, but there's also a sensitivity to specific individuals. So for EEG features to be relevant to neurodevelopment, we need to understand which features are very sensitive to brain maturation or atypical maturation, but also at a specific age, how the EEG features behave on interpersonal differences. So here, using a set of infant data of specifically 6 months of age, we try to understand which features were very stable and which ones were not that stable. So in fact, what we could see is that the complexity index, or the ones that we could connect with the complexity concept, were quite stable, as opposed to power and phase amplitude coupling, for example. So now that we know a little bit more about what those features actually refer to, I say a little bit, we can dig into what the different conditions that we know affect brain development actually from the start, how they affect also the brain signal. So assessing different genetic conditions, we can actually compare them, and we can try to dig into some of the genetic mechanisms that drive those genetic conditions. The other relevant aspect to this study, or these studies in general, is also that EEG can be done in animal models. And so finding translational features and translational task is highly relevant in this domain. So here I'll just show you a few examples of the different features and how they behave in different groups. So we see here that Fragile X syndrome, for example, has a lower alpha peak frequency compared to controls and ASD participants. However, both Fragile X and ASD have low gamma activity that is increased compared to controls. So this is just an example to show how different neuropathology from a genetic cause can sometimes on some features be highly similar and some other features may not be. The plethora of genetic conditions is really high and very diverse. So it's also relevant not to study really specific syndromes, but also study a realm of genetic conditions. And this is what we do when we assess copy number variation. So here I will be showing just a few slides on a data set that we've collected on 109 carriers. So these include 69 pathogenic CNVs. So these are heterogeneous children, basically, and they have a set of diagnoses, and they were compared to 250 controls. So just for example, what we see here on a normative model, so what we have in yellow the controls, and so even if they're put in a normative model, you can see the variability already in the controls. But when we add the duplication or the deletion, so our CNV groups, we see a decreased connectivity overall. And what's even more interesting here is that this decrease in connectivity is even enhanced when these children age. So this aspect of level of maturation and how EEG is sensitive to this is really relevant to longitudinal studies and trying really to understand the alteration that actually may grow with development in those populations. Another set of abnormalities, here we can see an increase in beta and gamma power, just like we saw in the ASD and Fragile X population. So this seemed to be a little bit constant across the different population we study. This was also the case in rare mutations in GAP1, where we saw in an older study this increase in beta gamma, to which I was very skeptical at the time. I wasn't sure if this activity was true. We have to remind us that these populations are not always easy to study because they do have a lot of comorbidities, and these SYNGAP mutation are really high risk for epilepsy, and therefore these children may have anti-epileptic medication. It was the case, of course, for those children that we've studied in this study. Benzodiazepine, as we know, may increase beta gamma, low gamma activity. It didn't seem to be the case in our sample, but still I was skeptical. So this took me really to collaborate with people working with animal models to try to have a clearer picture and not have that many confounds. So in the SYNGAP mouse, was epileptic. But of course, we didn't give anti-epileptic drug to the mouse. And we could assess the different features, how they behaved. And the different excitatory features that we could evaluate were high. For example, this steady state auditory evoked potential, very high in the SYNGAP mouse at 40 hertz and 10 hertz. There was indeed this low gamma increase in the mouse as well. So this was a confirmation of those results in humans. And we went on also to study the repetition effect and habituation in response to deviant. These are for clear reasons, given the translational potential of these tasks. And so in both the human and the mouse, we could see that there was a repetition effect in the controls, but not in the mouse. So this decision to actually study repetition effect and the novelty effect is certainly not novel. But it certainly gives some background to all of these studies that we're doing. So although the meaning of the response to repetition across species may not be exactly the same, it is still a translational task that we can use even in Drosophila. And in infants, the familiarity aspect is also something that has been studied for decades and decades and has been shown already behaviorally to be related to IQ. So since we do have a readout in EEG, I think it's a really relevant task to use in those kinds of studies when we do want to have a translational potential. So we went on to study habituation and novelty response in clinical populations and typically developing infants to understand how it actually develops. So in the genetic mutations that we studied, we did see alterations in the repetition effects. And here, for example, the SYNGAP and the tuberous sclerosis are really the ones that showed most anomalies. And they are quite contrasting with the response that we see in the Fragile X and in the Down syndrome. So although all of these syndromes do have big impact on brain development and induce intellectual disability for most individuals, these brain responses are not exactly the same. across the syndromes because of the differences in the molecular pathways that actually affect brain development. However, this is the habituation is preserved in neurofibromatosis, which is another genetic syndrome that can induce intellectual disability, but it's more learning impairments. So typically the impairments are less severe in this population. But we did see differences in the response to novelty, which really maybe is related actually to the ADHD phenotype. So really the ADHD symptoms in this population. In typically developing children, what we see is that the response is already there very early in development, but it shows a gradient with development. So we see here the first visit, the babies were 6 months old, but then so this nice response to repetition is there. The recovery for novelty is there, and this is a local recovery. It's a local deviant. But we see that with increasing age, 24 months, 4 years of age, the response is even stronger. So these responses are related to behavior, and this is of course very interesting. So in a series of papers, we've shown that these responses are predictive of the adaptive behavior at 6 months of age, for example. So at the same age, current age, but they can also have predictive capacity of the adaptive behavior at 4 years of age. So I think this is particularly relevant to the potential that we see in EEG as a predictor of later development. So when we continue with these kinds of oddball tasks, we see that it's not the same across the lifespan. So as we were able to predict adaptive behavior later in age from 6 months old EEG, when we went into the category of 4 years of age and older, we were not able to predict IQ per se, but we were able to predict working memory and processing speed. So just to summarize, I think what EEG can provide is certainly enormous. So I think it can help to understand the underlying mechanisms behind the alterations in the signals that we see. But for this, we need to continue to work with translational models. I think we can gain insight into the correlations that we have with behavior, trace, and diagnosis. And to pursue this, we need large clinical populations. I think it can facilitate early identification of atypical development, and for this, we need longitudinal studies. And I think it can be informative as outcome measured in clinical trials, and for this, we need clinical trials. So for these, I'm greatly involved in Q1K infrastructure initiative. So this is a large clinical study where we recruit participants and their families that have neurodevelopmental disorders, mainly autism, but it can be larger than that. These children have genetic mutation, but they may also have just a diagnosis. So this is ongoing at the moment, and we are excited to currently collect the data and share that data. And I'm also involved in several clinical trials. These include clinical trials in Fragile X, in autism, Rett syndrome, etc., and using EEG as a readout of the target of the medication. So with this, just want to thank my lab members. Oh, sorry.
Thank you, Sarah. Now we'll move on to Faisal, our final speaker, who is a member of the Global Brain Consortium, and we'll take it on to the Q&A after his talk.
Excellent. Okay, hopefully you can see my screen okay. So thanks everyone for sticking along. It's been an information-packed session, lots and lots of interesting sort of results. You'll be pleased to know this was a bit more light-hearted, less sort of empirical results, more interesting, hopefully sort of anecdotes, sort of an history of EEG. So as Sarah pointed out, it is 100 years almost exactly since the discovery of EEG. In fact, it's almost 44 days time, it will be the exact sort of anniversary, centenary of Hans Berger discovering the EEG in Jena, Germany. So many people know this fact. First thing he probably sort of introduced to EEG here, like it's a sort of a 1924, the birth of this sort of this field, I guess, but probably not many people know exactly how it went down. And so I have been digging through the archives of neuroscience history, mainly through YouTube, and I have uncovered a piece from National Geographic, which I think everybody will find of a substantial interest. So I'm about to unveil a fascinating video on the history, as Christine put it, the origin story of EEG. So here you go. I'm going to let it just play. No sound right now. I'll let you just watch and enjoy. Here's Hans Berger, of course. There he is with his multi-channel EEG. And it's sort of the birth of a supervillain, or the birth of a super signal, maybe. Okay, there we are. That is historically very accurate, as I'm sure you can sort of appreciate, imagine, right? And so let me just skip over. And so National Geographic there really sort of illustrating how sort of sensational this discovery was. And that was the kind of thing that sort of did not escape the press in the sort of 1930s, when it's finally sort of publicly sort of revealed by Hans Berger. And I've made sort of some excerpts here from sort of Cornelius Borch's amazing book on the cultural history of EEG. If you haven't read that, then you definitely should go and get a copy. So here's a set of letters to Hans Berger and some sort of press cuttings here on all the different sort of potential uses of EEG. And so we had, and I'll just sort of cut to the sort of chase, there were sort of three main sort of ideas. One was perhaps we could read the minds of others without having to sort of lift a finger. And then there was the consumer sort of applications. Perhaps we could evaluate whether one chair is more comfy than another, right? Or sort of the effects of sort of caffeine, sort of probably something that sort of is resonated sort of with some people in this sort of research that they'll have done. Consumer sort of evaluations, neuromarketing as it's sort of called these days. And then perhaps most importantly of all, can we measure the temperament of a horse using EEG? And so these are some of the applications that were put to Hans Berger. And the sort of general idea is, hey, look, this is finally, we found an electrical record of thoughts, and it really sort of gripped the imagination of the general public. The scientific community was slightly more skeptical. And so it took a few more years for it to sort of get hold in the sort of scientific, or the neuroscience, the sort of nascent neuroscience sort of community. And that was largely driven by here Lord Douglas Adrian, who was a Nobel laureate with Sir Charles Sherrington a few years sort of prior. So what Adrian and his colleague Brian Matthews did is validated this work. So they were skeptical, and what they decided to do was repeat his experiments, repeat sort of Hans Berger's sort of work. And so after an initial period of hesitation, they confirmed that, hey, look, we can also find the same pattern of results that Hans Berger has. And so classic sort of alpha suppression sort of signal, eyes open versus eyes shut here, which they called the Berger rhythm. And so a lot has happened since Adrian and Matthews' replications. And so one of my colleagues, Dr. Richard Nahr, has put together an interactive timeline. I'll share this link, we'll share the slides with you all so you can explore the sort of history of EEG all the way up until today. So that's sort of where we are. We've made a lot of progress in that time. Like sort of Sarah was showing some excellent sort of research recently on some potential clinical utility of the sort of signal. But look, we also have horse temperament now, right? There are commercial applications that can track sort of EEG in horses. Of course, we also have sort of neuromarketing. Sort of there's a lot of sort of applications here as well with EEG. Still relatively in its infancy. Probably a lot sort of a way to sort of go there, but it's developing. And so we said, okay, well, what can we do to sort of commemorate this incredible sort of moment in history? 100 years almost exactly since the discovery. And we put our heads together. This is sort of with the EEGNet team, the Global Brain Consortium, and sort of EEG main labs groups, the coordinating sort of groups for those three initiatives. And we said, okay, well, what can we do? So we said, what we'd like to do is take a snapshot in time. So let's see, what do people in the neuroscience community, broader than just sort of the EEG community, what do people think of sort of EEG right now? What do they think it might look like in the next 10 years and the next 100 years? And sort of save that sort of for posterity, sort of put that on the scientific record so that future people, future historians of science or sort of neuroscientists might look back on this. Maybe they'll laugh at us at being sort of so optimistic on some of our sort of predictions, but this is the general sort of idea. So we sent out a sort of survey. Some of you on this conference call may well have even completed this survey. So thank you for that. We got a large number of sort of responses, significant amount sort of EEG experience amongst those sort of people. And so everything I'll show you now is all sort of going to be publicly available. The survey, if you haven't completed it, we're still collecting more and more sort of responses. You can find that in the link below. And so what did we find? Well, so I'm going to show you this sort of a list here. So the reason why what we did is we generated a list of statements of where EEG might be applied. And so we came up, I'll show you in the next slide, it's probably a bit cleaner. We came up with a list of statements, some of those where we believe that EEG is already being used in some areas, and then other statements which we really felt were probably impossible beyond the sort of realm of sort of EEG sort of capabilities. And then we asked people to put a number against this. When do you think this statement will be fully realized, and there'll be a sort of scientific consensus that this is something that we can do with EEG? So we gave this list of statements, we asked people to rate them, and then what you're seeing here is just a sort of ordered by nearest to furthest away. So the idea that EEG can be routinely used in diagnosis and monitoring of sleep disorders, people suggested, yes, it's very, very soon. So in fact, in many quarters saying it's already here. We had others here, like, so we have high quality EEG data can be acquired from everyone. Well, actually, perhaps a little bit further off, maybe there's some additional sort of technological sort of developments that are required here. And I'm not going to go through all of these sort of 30-odd sort of statements, but you get a feel for where we are as a sort of a EEG sort of community, and that's sort of broad sort of terms from research, clinical, from industry, there's sort of tech development, software underpinning that, and so on. And then we come all the way towards the end of things that we feel as a community are much, much further away. So widely used as a lie detector, and so and relevant for us as sort of in terms of the Global Brain Consortium and various other sort of international sort of initiatives, widely used across an indicator of brain health. So questions around why do people sort of feel that is something that we'll be probing into. And then we had two really off the wall sort of ideas, whether EEG might be used to sort of read the contents of dreams or the contents of your long-term memories. And so this provides a really nice sort of fun sort of picture. I'm pretty sure almost all of these predictions will end up being sort of incorrect. The question is sort of to what degree, and also what does this tell us about people's perceptions right now? And so then we also came on, so that was a bit of fun. Okay, I'll just make some sort of speculations. I also came on and asked some sort of serious work-related sort of questions more immediately. So we came up with a list of sort of elements of sort of research and sort of application, different sort of parts of that sort of pipeline. And we said, where would sort of developments really lead to a step change in your working practices? What should we be prioritizing collectively as a community to drive forwards innovations and in sort of a impact of EEG? And so what you see here are as a sort of list. Again, it's ordered based on sort of how important the sort of respondents felt these sort of areas were. And you can see really interestingly, artifact detection and removal methods, the most popular. This is where the sort of EEG community is saying this is what we really need to keep sort of improving on. We have others around data acquisition, accuracy of source localization. These things are sort of big priorities for the sort of community. And so together, this with our sort of predictions, it gives us sort of a blueprint of where we might sort of go in the future. And so potentially sort of informative for us in the next sort of few years. And this sort of centenary has also sort of captured the imagination again of the sort of popular press. So we have sort of colleagues in Jena who are running an event on exactly the 6th of July. So this is a sort of press cutting that just came out a couple of weeks ago in a popular sort of magazine in Germany. And they are running a recreation of the original work in this sort of laboratory. And they've been looking at getting hold of some of the very old equipment that's available in Jena and sort of getting it to sort of function and perform some recordings. Of sort of colleagues Barbara Schmidt and Doreen Barloff who are running that. So there'll be a public lecture series there to sort of commemorate that sort of centenary with some well-renowned sort of EEG scientists. So Pedro, who you heard from earlier, Pedro and Professor Dezhong Yao in Chengdu in China are also sort of commemorating this in a slightly different way, running a sort of a conference, the first ever Belt and Road conference on sort of brain health, which will be running in sort of September. And so I'm sure we'll be sharing more information about this as this sort of as the details get confirmed. But perhaps we'll see some of you over there in China on a sort of a set of symposia across various different sort of application areas of EEG relating to brain health. And then finally, another sort of event to commemorate the centenary is with sort of cutting EEG. Some of you will know a sort of a largely sort of European focused sort of conference on sort of methods in EEG, state-of-the-art sort of techniques. And so in October, we will be launching a sort of manifesto, which sort of builds on the sort of results that I've just sort of shown you, which is really a call to action and a sort of public commitment to pursuing various different sort of priority areas identified through our sort of survey, and to then sort of help promote its application across various different sort of domains. So we'll be sharing more about that as it sort of develops, but it'll be publicly sort of announced in October at the Cutting EEG conference, which is also online as well as in person. And so finally, this is sort of a request for some support from people here. So just like sort of Adrian and Matthews were skeptical about sort of the EEG sort of discovery by Hans Berger and had to go and do it sort of themselves. This is something that we've been doing with EEG Many Labs, which is we've been replicating 20 of the most influential EEG studies. And what we're asking the community now is to vote on which of these results that we are replicating will actually sort of succeed. And through that, we hope to be able to understand what are some of the sort of key features that make EEG science sort of credible. And so we've created a sort of survey that goes through all of our sort of studies in partnership with some colleagues in sort of Sweden. And we're asking you to sort of, hopefully, if you can sort of spare an hour of your time to complete this survey, you will earn some money immediately for a brain health charity. And also, if you are successful with your sort of predictions and you make some sort of good sort of bets through our prediction markets that we're running linked to this, you'll also be able to earn up to $300 US for either yourself or for a charity. So hopefully it's worthwhile effort, and in that process you'll also inform us about the credibility of various EEG phenomena. So I'll wrap up there. and say thank you very much for listening, but also thank you very much to all the sort of colleagues who have helped in the development of this sort of this survey. So there's too many here to sort of go through, but it's on that list there. So thank you. I will end there.
Thank you very much, Faisal. We will move on to the Q&A in just a moment. I just wanted to remind people if perhaps we could get a slide up. Thank you so much. So we're just to remind people that as we move into the Q&A portion, please do put your questions into the chat and we'll take them from there with Alan moderating. And to recap, to brief mentions from the presentations, if you'd like to get involved with EEGNet and request an account, please go to eegnetloris.ca. You'll be asked to review the terms of use, reset your password, and we would also recommend requesting a CBrain account. And furthermore, if you'd like to ever contact or get support from the EEGNet team, here's how to reach us or scan in the QR code. And we look forward to hearing from you. So with that, thank you to all of our speakers today for presenting a variety of corners of the EEGNet grant, as well as the local and global ecosystem, certainly the history. And with that, over to Alan for the Q&A. Please, we invite your questions at this time.
Thank you very much, Christine. And thank you very much to all of the speakers. This is, as I've often said, this is taking a drink from a fire hose to try and keep up with all of that has been presented today in the EEGNet colloquium. We've heard methods and applications, meetings, networks, new initiatives, the entire landscape of EEG globally and indeed nationally. So I think it's been a very successful session. All of this has been mediated by the CONP infrastructure, so we can thank Brain Canada for all of their support in making this happen. I'd particularly like to thank Christine Rogers, Irem Katitas, Derek Lowe, and Jefferson Casimir, the EEGNet team, for all of the sterling work in putting this colloquium on and making sure that it ran smoothly. So thank you very much, team. It was great. And the slides, some of the speakers are happy to make their slides available. We'll follow up with the individual speakers to see if anybody else wants to share their slides. If you are interested in a particular slide deck, please get in touch with our team, and we will field and manage the inquiries for you. With that, we'll now go on to the Q&A session. Please put your questions on in the chat, and I'll try and monitor and redirect those questions appropriately. So
I think for starters, we'd like to invite perhaps views from the audience as to the future of EEG. Just looking at these applications that we have going forward with the platform, are there any comments, particularly relating to the 100 Years of EEG survey, as to where this could go in addition?
So Christine, was that a question to me? Is that what you were asking?
Yeah, perhaps.
Yeah.
Sure, for any of our contributors, yes. Yeah,
okay. Well, I'll I'll go with this if we're waiting on some of the questions. So I think this kind of platform that EEGNet provides is really, I think, very much needed for the community. It's something that came out across our different questions, and something I didn't go into here, but can be accessed online, or the open-ended responses that people provided. And so this is a resource like this. I can see myself making use of it. Our team will, I'm sure, over the coming years. But also it helps us democratize EEG. I know this is a passion of your team over there in Canada, to be able to make this publicly available. And so I think this is the direction of travel that is much needed, and hopefully will lead to new discoveries that are beyond the abilities of any one individual group, and you need a diversity of expertise to come at it. And this is the type of platform that allows that. So I'm really excited about the future, thanks to initiatives like this.
I have a question for Philippe and or Bosch. And that is that as you put these tools on CBrain, there are two aspects to potential advantage. One is accessibility. You can actually get at these tools and you can try them from anywhere in the world. But secondly, is there any speed up in the actual processing, or is it simply just about getting access to those tools? So Philippe, perhaps.
Yes, especially if that's what I was trying to insist on this aspect, that the access to the tool is really important. But if you have access to a large data set, and if you want to run that locally, it's going to take forever. So one of the main advantage is really the computing resources that you can benefit from CBrain. So it's really access to the tool, but also the computing resources to speed up everything.
Do you have a sense of how much of a speed up you might expect? Is it like two times faster, or 10 times faster? I understand it depends on what application you're using. Do you have a feel for this?
I think I can ask Corentin if he's still there.
Yes, I'm still here. Do you hear me?
Okay. Yeah. Do you have an idea about how much it would speed up the processing? Example, if
I mean, I don't have an exact number because it depends on where you're starting from, what's your local resources, but it's definitely a big advantage. But it's hard to just put a number on that.
But you can parallel processes, no? On CBrain.
Yes, you can. And that for, like you mentioned, for 100 subjects, that's definitely going to be more effective.
And I think part of the efficiencies that you already presented is in seeking the packages and the exact configuration and all the tooling around getting access, around setting up the entire pipeline for processing. Really, it sets up a situation where users can enter their parameters and add their data and have it be processed in a very efficient, standardized way on the national infrastructure, and reducing significantly the technical burden needed to run these analyses. So I think the acceleration there for researchers, I'm not sure we can even measure it, frankly. Brian might have a better idea as to how the containerization on HBCD does accelerate the process. I'm not sure.
Yeah. I'm just mentioning quickly from. Sorry, go ahead, Jorge. You go first, please.
I can say that a process that I started in my computer of the harmonized QEEG that was taking 2 days in my computer was finished in less than 12 hours by Arioski in Chendo in the CBrain copy we have in Chendo at Peter's lab. So he finished the same analysis I was doing in less than 12 hours. And it's not the same cluster and computer resources. So in Canada, there are much power.
Yeah, and this is important if you're in low and middle income countries globally, because oftentimes you don't have the resources locally. So getting access to these either high performance computing or cloud services makes the difference, not just in speed up, but actually feasibility at all of doing the work. But yeah, questions are starting to come in. So we have a question from Priyam Kar. What are the tools for source localization based on EEG on EEGNet platform, especially related to source connectivity analysis?
I can describe what is running right now. So we have many for source reconstruction. We have many tools that have been developed by Philippe and Brainstorm, and you can run all of that on CBrain. So for the source analysis, you can do minimum norm with constrained orientation for the source. You can do the beamformers of Philippe, also for the frequency analysis. In terms then of connectivity, once you have performed your sources, so you can perform the source for the Hilbert transform or for the evoked response or for the raw data, or for the given specific frequencies using field trip functions. Then for the connectivity is quite variable, but it's more common tools. So you can extract n by n or 1 by n coherence phase locking value, Granger causality, phase transfer entropy, envelope correlation. I have a other list, so you can. So it's quite well. We have many tools for that, and you can do it on different regions of interest, or you can do it whole brain. You can do it etc. In fMRI, we would say voxel, seed to voxel, but you can do the same thing with the vertices if you are working on the cortical mesh. So all these type of tools are already available, and you can run them directly on CBrain just with one pipeline.
So please do follow up with our team so that we can redirect these questions and you can get a complete follow-up and perhaps continue these discussions in more detail offline. The next question, Vasily Vakorin asks, a question about routine clinical EEG. If such data are available for research, what kind of corresponding information should be made available, prioritized for release, along with EEG records themselves?